Bioremediation is defined as the process where organic waste is degraded in the controlled condition into the levels below concentration limits established by regulatory authorities with the help of microorganisms. This process is invented by George Robinson. The microorganisms used for this process are bacteria, fungi, and algae. Bioremediation is an option that offers possibility to destroy various contaminants using natural biological activity. The oxidation reduction potential in soil and groundwater as well as pH, temperature, oxygen content and breakdown product concentrations are used to monitor bioremediation process indirectly. The main requirements for the growth and metabolism of the microbes is energy and the carbon source. They get this from the waste or the contaminants and utilizing that the microbes perform degradation. For this purpose, the enzymes or metabolites of the microbes are used. Organisms attack enzymatically on the pollutants in order to degrade those pollutants. Microbes get adapted with the system and remediate the environmental hazards. Factors involved are microbial population and environmental factors. The microbes will get adapted to the environment and due to the adaptability, they will grow and degrade the environmental contaminants. Environmental factors include nutrients availability. So, if the energy source, that is carbon, is present, the organism will grow. Similarly, in the presence of suitable pH and temperature, the organism will grow. Category of microorganism used in the process. First one is aerobic. Aerobic means organism which require oxygen for their growth. For example, Pseudomonas, Mycobacterium, Rhodococcus, etc. Anaerobes do not require oxygen for their growth. These are not frequently used, but sometimes used in decontamination of river sediments. The third one is lignolytic fungi, which are white rot fungus. Methylotropes, these are aerobic bacteria that grow utilizing methane for carbon and energy, and hence they are called as methylotropes. Techniques used for the bioremediation process. First one is in situ bioremediation. It is biological treatment of contaminants in the subsurface, typically in groundwater or soil. The most important in situ treatments are bioventing, which means supplying air and nutrients in the contaminated soil. In situ biodegradation, also oxygen and nutrients are provided. Biosparging is the process where air is injected in the groundwater. Bioaugmentation is the addition of microbes at the contaminated areas in order to decontaminate. Second one is ex situ bioremediation. Land farming is a technique in which soil is sprayed on the soil bed till the pollutants get reduced. Second is composting, which is combination of agricultural waste and contaminated soil, which gives rise to microbial population which degrades the waste. Biopiles is basically combination of the two techniques, that is land farming and second one is composting. Bioreactor is a vessel which is able to carry out degradation of targeted compounds. Advantages of bioremediation. First is environment friendly, second is safe, third is low energy consumption, and fourth is removal of harmful compounds. Disadvantages. This process is very much time consuming, and second is only biodegradable compounds are degraded by this process. Latest updates. A novel bioremediation techniques was found by National Institute of Ocean Technology, that is NIOT. It is a bioinformatics tool named RemiDB. This would help to identify novel bioremediation related enzymes from a huge set of metagenomic sequence data. They have isolated bacteria from sludge with 1,4-dioxane, which is carcinogen. This bacteria has enzyme which converts 1,4-dioxane into harmless CO2 and water. The bacteria is Pseudonocardia dioxynivorans, and hence the bioremediation is achieved in this case.